you are audible now.
A very warm welcome to everyone joining us for Acchutha Inspired Series 17. It's my pleasure to introduce our guest, Dr. Charanya. Dr. Charanya completed a Bachelor of Optometry from Allied School and earned a PhD from Indiana University. She's currently a scientist at Brien Holden Eye Research Center. We are truly honored to have us with us here, ma'am. Our topic is about transition from clinician to a basic science researcher. Please join me in giving a warm welcome to Dr. Charanya. Over to you, ma'am.
Okay. Am I audible?
Yes, ma'am.
Okay. All right. Thank you for that kind introduction, Shamala. And I should also thank Gopinath and Saranya for inviting me to share my uh, journey, I guess, where I transitioned from a clinician to basic researcher. Thanks to both of you for initiating this series and having me a part of it and giving me an opportunity to hopefully inspire a few to probably take this route. Okay, so without further ado, I will get started. So I thought I will sort of give a background of how my career has sort of gone from India to the US and back. So I did my undergraduate training in optometry from Elite School of Optometry, and Saranya is my senior. We were scared of her quite a bit. But so after my undergrad training from Elite School, I actually went to do a PhD in Indiana University, Bloomington. And this is where I sort of forked away from optometry and got further into, hardcore cell biology. And after 6 years of training there, though I wanted to do a postdoctoral training in the US, I came back to India, this time further north of Chennai. We came to Hyderabad. And since 2010, when I joined the LV Prasad Eye Institute as a postdoctoral fellow, until now, I am, currently working in the same place. So I did my postdoctoral training for 3 years, after which I joined LVP as a research scientist. So I've been asked to talk about, my transition from a clinician to a researcher. Now, it's a very, very broad topic, and it's a little difficult to sort of put it in, a gist without boring you guys. So I thought I will, uh, partition this journey in four parts: my undergraduate days, graduate days, postdoctoral fellowship days, and now as an independent scientist. I will share with you a little bit about the work that I have done at each stage of my career and the learning that has sort of shaped me to be where I am from each of these stages. All right. So the first one, of course, the undergraduate days. This was basically bliss. Friends, some studies, yes, a lot of clinical training, yes. And so the question was, why did I choose optometry? So some people might have chosen optometry because they have a family, an optical shop or an optometry practice. I came into optometry by elimination. All right. So I did not want to do medicine. I did not want to do engineering. That much I was clear about. So that left me with choices such as your basic sciences, physics, chemistry, zoology, biochemistry, etc. And then there were these options of physiotherapy, law, and I hadn't heard of optometry at that time. So it was again by chance that someone mentioned optometry, and my immediate senior, who also happens to be a distant cousin of mine, was in elite school studying optometry program. She was in the optometry program. So it was more out of curiosity. I frankly did not know what optometry meant. So I came, gave the entrance exam, sat through the interview, got an admission, and I loved it. Elite school probably gave us the best year, 4 years, both in terms of training, because we got into the clinic in the second year. We tagged along with our seniors. So we were able to right away apply the theoretical knowledge that we were getting in the classrooms to the patients. So it was wonderful. I really enjoyed it. And the last year of internship um, taught me that I am not ready for clinic, in the sense clinic is not my cup of tea. It was a little, the very fact that I did not know what happened to my patient at the end of the day did not settle right with me. And so I said, you know what, this is great. I mean, I love that I'm able to help so many people. I feel bad that I'm unable to help a lot of people. However, this is not something I want to do. So again, by elimination, uh, I chose to do research. So this is what most of our seniors did. Many of them went abroad for a PhD or to do an OD program. And since I knew I was not going to be in the clinic, I chose to go the route of research. And the initial foray into research through the third year project, as well as when I was in I think second or third year, Dr. Michael Webster was visiting, and I got to see firsthand how a research question is asked and how you meticulously answer that question by using very simple tools, how to gather data, etc. So that was very interesting to me. And so I decided that is what I want to pursue. So after my undergraduate degree, I applied to several universities: Ohio State, Indiana University, to University of Waterloo, um, and I got accepted into Indiana University. So through my undergraduate training, the biggest learnings are, you don't need to necessarily know exactly what you want to do. There are people who know exactly what they want to do. They come guns blazing. I was not one of them. So choice by elimination is absolutely acceptable. But whatever you choose to do, enjoy doing it. And I also would say that every opportunity that is available to you, you should use that to build your skills. So, for example, every summer I used to go and volunteer in a public health service center in Mambalam. And that is where I really learned to do refraction. There was one optometrist who could just swing the retinoscope once, scope, put a lens, tree, and prescribe glasses like that. And that was the first place I also learned how to use a Shire stone. So utilize every opportunity that is available to you to build your skills. So at the end of 4 years, my retinoscopy skills were really good. Um, and also use every opportunity to sample different areas. For example, I again, after my undergraduate degree, in the interim period between me finishing and joining my graduate studies, I actually volunteered for a glaucoma study. So I used to work with Ramesh SVE, and we were collecting data on patients who had glaucoma using certain instruments, and studying progression. So again, that was a very useful tool, because it once again taught me how to ask a question, how to meticulously address a question without rushing through any of the processes. We went through the whole notion, and those 6 months were very useful because it helped me learn a lot of new skills, and most importantly, learn patience. And of course, you make friends for life. And even today, we get together, we enjoy the picture on the bottom was taken, I think, during EWOC this year. And so we are thick friends. We are friends for life. We enjoyed our 4 years, and there was a lot of learning. So my transition from a clinician to a researcher was very quick. I never once imagined that I will actually get into doing very basic biology, cell biology. But as fate would have it, when I applied to multiple universities, I actually got admission into Waterloo. However, thankfully, Canada requires you to have a medical checkup prior to the visa processing, and it turned out that I had tuberculosis. So, very atypical, no symptoms, absolutely. But that medical checkup helped us, diagnose that this problem existed. So, I had to defer, and eventually, I had to take a year break to recover from the treatment. And so I reapplied and I got admits in Indiana University with Dr. S.P. Srinivas. So the person on your left in the white shirt, he's my boss, my guru who, sort of led me into this world of cell biology. So I remember the first day I went into the lab, he gave me a bundle of 10 papers and he said, here, read these papers. Our lab sort of focuses on this. Now, we did do a third year project. We were not very good at reading papers or understanding the papers. So here I am, who doesn't know how to read papers, leave alone how to understand or to debunk your protein interactions. Now I know what proteins are, I know what genes are. That's pretty much all I know. How do they talk to each other? Why do they talk to each other? How do they influence each other? Now this is all way beyond, right? So I used to spend um, hours, probably days, reading the same paper again and again and again until I could understand the words. So first I had to start with understanding words. Then I had to, sort of put it all together to understand what the paper was conveying, right? So our coursework, which we did in the Department of Biology, it was amazing training. So um, we were asked to pick up a paper, and each one of us had to discuss only one figure in the paper. And for you to, sort of discuss that figure, you had to read the entire paper and understand. The other exercise that we did was we were given the paper and we had to write only the abstract. So the abstract was removed. We were given the body of the paper and we had to reconstruct the abstract. So these were extremely challenging, very difficult. There were many days when I asked myself, why am I doing this? And, I wasn't able to see the light at the end of the tunnel initially, but I kept through and I persisted. And it turned out to be, real amazing lessons for life, which now I torture my students with. So I thought instead of me generally talking only about my journey in terms of, the philosophical angle, I thought I will also give a glimpse of the work that I did, especially because the pictures are very pretty. We use colors. One part of the my PhD thesis was focused on understanding how the actin myosin. So you guys might have heard of this term, actin and myosin, which are the two proteins that help the muscle fibers sort of contract, and that is what causes the muscle to, that is what makes the muscle contract, right? So the same system, the same proteins exist in other cells also. So not only in muscle cells, but also in other cells. So our interest, my lab, my boss's lab, was primarily focused on understanding how this actin and myosin interaction, how does it help maintain the barrier function of endothelial cells. We were also interested from the angle of trabecular meshwork cells in terms of when the contraction occurs. And if you can imagine the trabecular meshwork to be like a sponge, so if you squeeze the sponge, then the porosity reduces. When the porosity reduces, the resistance increases, which means the intraocular pressure will increase because less aqueous humor is flowing out. So we were actually interested in understanding how this contraction of these cells might regulate aqueous humor outflow as well as endothelial permeability. So I will sort of talk a little bit about the endothelial work that we did. So you all know that the corneal edema can occur because of various reasons. It can be trauma, it can be Fuchs dystrophy or PPCD. It can be because of inflammation. Our interest was primarily inflammation, which occurs, say, after a cataract surgery. And that causes the endothelial cells to become more permeable, and that causes the corneal edema or the stromal edema. So bear with me. This is something I do teach my undergraduate students. So my course is a little tough for them. So what happens is basically two cells are held together, and depending on how close they are held together, um, you can determine what goes through in between them, right? So what holds the cells together? Okay, what determines how tight this junction is? There are proteins called tight junctions. As the name suggests, they basically determine what is the distance between two cell membrane. If it is too small, then we call it as a junction which is less permeable. If they are not too close, then it's a more permeable junction. So the endothelial cells, say for example, compared to corneal epithelial cells, are more permeable in general because they have to allow aqueous or the nutrition to reach the stroma and then remove the waste. So in general, they are more leaky. However, if you now disrupt this tight junction, then all hell breaks loose, and absolutely there is no control over stromal edema. So this is the sort of the idea that we are working with, and we wanted to understand how the actin cytoskeleton. So what you are seeing here is in red. What you are seeing are these nice fibers. These are the actin fibers, much like what you would see in the muscle. So you are seeing the actin fibers, and you are also seeing the tight junction. One example, one protein that is found in the tight junction is the ZO1 protein, which we commonly study as a surrogate for in the permeability. So you can see how beautifully in the corneal endothelial cells, the ZO1 or the tight junction protein is arranged. And the actin filaments, if you see, they go in circles. Okay. So they actually connect and they form like a scaffold. So they are very important for maintaining the proteins at the cell membrane. So if the actin cytoskeleton contracts, then it will pull apart the proteins, increasing the intercellular permeability. So this is what we were working with. This was the hypothesis that we were working with. So as long as the contractile forces and the tethering forces were balanced, you're going to have barrier integrity. When there is a mismatch or an imbalance, then that is going to lead to barrier dysfunction and endothelial or stromal edema. Right, so we used certain agents which are released during inflammation because that was our model of interest. And you can see in control, meaning when the cells are not disrupted, this is their baseline resting level. You can see how beautifully the actin filaments are arranged around the periphery of the cells. But when we treat them with a drug that is known to disrupt, that is known to cause contraction, you can see how angry the cells look. The actin filaments are really contracted, meaning they are bunched up, and you can see the cell size has reduced and the space between the cells has, so you can see that they are literally tearing apart the cells. So this is what happens when there is a barrier breakdown, and that can cause the corneal edema. So our interest ideally was to find out, okay, this happens, how do we prevent it from happening? So now, if you prevent the actin from contracting, ideally, then you should prevent the breakdown. So we were working with a few compounds. You guys might have heard, now it has become very famous, the Rho kinase inhibitor, Y27632, or it is commercially available as Ripasudil. This drug right now is being used to treat or reduce IOP in glaucomatous individuals. However, it works fine with the corneal endothelium as well. So we used Y, which is a Rho kinase inhibitor, So with thrombin, you can see the barrier. So what we did was we grew the cells, we added a dye and a protein of a certain a size. Okay, so normally that protein cannot pass through between the cells, but when you pull the cells apart, the protein can go through. So an increase with thrombin tells us that the protein is going through, that the intercellular permeability has increased. When you treat it with the rho kinase inhibitor, which actually prevents the contraction, it loosens the actin cytoskeleton so it cannot contract, then you see that it negates the increased paracellular permeability or intracellular permeability that occurs in the presence of thrombin. Okay, so I hope this was not too overboard. However, I just wanted to sort of give a sense of where I was, from optometry training, looking at patients' eyes, looking at the endothelium through a specular microscope, to actually going and studying what happens and what is the mechanism behind it, right? So this was, like I said, extremely tiresome. It was extremely challenging, uh, but I learned a lot. And, it was absolutely wonderful that I got this opportunity to do. So, in all of this, my learning through my PhD was that you have to work very hard and persistence. You need to be extremely persistent. There are going to be days or weeks where you are like, why am I doing this? But you keep at it and you will see the light at the end of the tunnel. And it is absolutely okay for you not to know everything, because as a clinician, we are taught to put that facade in front of the patient to say that I know everything, I'm in control. We do that for a reason. But in research, you really have to break that facade and you will have to accept that I do not know this. And that's absolutely fine, because we truly do not know. Um, this was very difficult, but it was doable. So if you are planning to, sort of digress from mainstream optometry, go for it. It will be that much more challenging, but it will be worth it at the end. And the best way to learn is to do it, right? So I had to learn how to pipe it. I had to learn how to dissect. I had to learn how to pull out a trabecular meshwork, chop it up, put it, prepare medium, the nutrition supplements that we give to grow the cells, etc. I had to learn how to, do different techniques which I've never heard of in my life. And the best way to learn all of that is to help someone do it. So the postdoc in our lab, I used to sort of, harass her, and I used to tell her, I will do this part for you. I will do that part for you. Let me make the gel for you. Let me prepare the sample for you. So that's the best way to learn. And there is no dignity in labor. I also used to fix a lot of things around in the lab because America is a land of do-it-yourselves. Unlike India, where you just call a person and somebody is at your beck and call fixing things for you. Over there, you had to learn to, what do you say, tinker around, fix things, and yeah, that was wonderful. And I mean, this is something that many of us talk about is that PhD is more philosophical than scientific. Actually, it is a doctor of philosophy for a reason, because it teaches you how to be humble. It teaches you to accept that there are many things that you do not know. It also teaches you to really sit down and think and introspect, which is what philosophical training is largely about. And it hinges heavily on critical thinking and logic. And since I love puzzles, this actually was very useful for me in the sense that I really like to sit with a problem and get to the guts of it. And because I was surrounded by a bunch of optical engineers for fellow graduate students, I had to learn to present my thoughts in simple terms, which meant that you really, really had to sit with your data, understand what it says, and then present it in simple terms. So these were the learning for me during my graduate studies. All right. So following that, I came to LV Prasad Eye Institute. I joined in 2010 as a postdoctoral fellow. It so happened that they were looking for a person to do some translational research. So this is a new area of research for me. I got indoctrined into stem cell research, which was very exciting because that's the buzzword. And the translational research is basically, you can imagine as you sandwiched between a basic scientist and a clinician. So you are a person who understands both languages now, and you are able to, sort of address both ends of the requirement. So which is precisely what I was doing. So I was introduced to limbal stem cells. So limbus, as you all know, lies between the conjunctiva and the cornea, and the palisades of Vogt is where the stem cells for the corneal epithelium is located, right? So when there is, chemical or thermal burns or aniridia or Steven-Johnson syndrome, the stem cells or the palisades can get affected, leading to conjunctivalization of the cornea, also chronic inflammation, epithelial breakdown, pain, vascularization, etc. So what people have been doing is there is a lot of treatment strategies, right from taking a part of the limbus from either a donor or from the other eye, contralateral eye, and grafting the whole limbus along with the conjunctiva, or people have taken conjunctival cells and then tried to grow the epithelial cells. And then there are studies which actually just took the limbus either from a donor or a living relative or a the contralateral eye which was healthy, a small fragment of it, and then they grew the corneal epithelial cells out of it. So which is what we did, and this was our project. So the major benefits compared to using the part of the limbus from the other eye or a donor was you use less tissue, which means that the chances of deficiency in the contralateral eye is heavily reduced. There was faster epithelialization, faster recovery of the ocular surface, less scarring, and you are amplifying the stem cells before transplantation. So the way it is done is, let's say in this patient, one eye is affected, the other eye is normal. So you take 2 millimeters or 1 millimeter of the limbal region from the eye which is normal, and then you chop it up into small pieces, okay, which is what you are seeing in panel 3. And then you put it on a human amniotic membrane, which is what you are seeing over here. And then the cells start growing out of the limbal tissue. And by around 14 to 20 days, you have a nice monolayer of epithelial cells along with the stem cells. So what the surgeons were doing is basically taking this tissue and they were grafting it on the affected eye. So some patients would not require a penetrating keratoplasty, but some where there is stromal scarring, you then do a PK and then, restore vision in that individual. So in terms of clinical outcomes, this was doing wonderfully well, nearly 70% survival rates. And we had at LV Prasad, we had one of the largest patient pool. We had done this in around 800 patients and around 70% success, which is amazing for a stem cell transplantation. So this is one of the other tissues or stem cell transplantations apart from bone marrow that has been successful in the clinic, right? So you can see how beautifully before the surgery and after surgery, how the recovery has occurred in this particular patient, right? So what we were working on for my postdoc was we wanted to develop a scaffold. So not only was I introduced into stem cell into the world of stem cells, but I was also getting into the world of materials. So I was sort of combining these two, which were totally two new monsters that I was dealing with. So why did we need to develop a scaffold? So we were using amniotic membrane. First of all, human amniotic membrane is not widely available. Apparently, in the UK, there is a dearth for human amniotic membrane. and our collaborators were from the UK. Obviously, the thickness, the quality, the degradation time, this is all very variable. It varies from donor to donor. It has a very tedious processing. So you have to remove the amnion from the chorion, test the donor for various pathogens. So it makes it also more expensive. And you have to keep it frozen. So that means that the cold chain has to be maintained. So we wanted to develop a material that is a synthetic material. Okay, so it is polymer based. So which means that you are not looking at a biological material. Since you are making the material, it can be made of a consistent thickness, quality, degradation time, whether you want it for 4 weeks, 6 weeks, 8 weeks, it's up to you. We can also obviously bulk manufacture it, store it at room temperature, and we were planning to do it as single pack, like intraocular lenses, right? So we actually did this. We went from manufacturing it, testing it in the lab, getting the regulatory approval, doing a preclinical trial in animals, and taking it to human trial all in under 6 years, including the human trial, right? So we did the manufacturing of the material, which you can see over here. This is called as PLGA, or polylactic and glycolic acid material. So lactic acid, you guys would have heard of, that's the chemical that we produce when we use our muscles too much. So that's an end byproduct of your your glycolysis cycle. And so it's a very inert material, easily absorbed by the body, with no immune response elicited. So in vitro testing, we could see that the cells that we were growing, we had amniotic membrane as a control. So the cells were supported, stem cells were growing, they were happy on the material. And then we had to take it from so it's one thing to work in the lab, and it's another thing to take it to the clinic. So whatever you do in the lab, you have to now put it through what we call as a GMP, or good manufacturing practice. These are sterile environments where your material has to be produced, has to be packaged. And then we took it to perform safety trials in rabbits, because that's a prerequisite before you go to humans. You have to do a safety trial. So in a safety trial, you're typically looking to see whether the material is safe, whether it is eliciting any adverse reaction or not. So we did this trial in rabbits. We saw that the material was degrading, no immune response. The rabbits were happy. And then we went to human trials. So we did it in 5 patients. And you can see the pre-op pictures and the post-op pictures. You can see a little bit of the material on some of the eyes at about a week. And by 3 months, the material is completely degraded, no immune response. And in many of them, the ocular surface was clear and smooth. Some of them obviously required PK to restore vision. So this was one project that actually took me, or gave me, an opportunity to work in the laboratory, learn about the regulatory requirements, and take the product to the clinic. So we went the full circle from lab to the clinic. So bench to bedside is what we did. One of the things that happened during a conversation that we had with our collaborators. So we were talking about, how can we do this better? And so we said, do we really require this material? Can we not directly put the little explants onto the ocular surface, on the amniotic membrane, and use the body as the incubator? So this was a thought experiment. Somebody blurted out their thoughts in our meeting. And the clinician in charge at that time, Dr. Virender Sangwan, decided that he is going to do that. So he came up with this entire technique of simple limbal epithelial transplantation, wherein you simply take a biopsy from the donor eye, and then you clean up the recipient eye, which is affected with LSCD. You place an amniotic membrane on the affected eye, and then you take this biopsy, you chop it up into small pieces. Instead of putting it on the amniotic membrane and keeping it in an incubator in the laboratory, you directly put it on the patient's eye. You put a bandage contact lens and you leave it. So you can actually see this is post-operative 1 month. So you can see the explants that are on the amniotic membrane that have been placed on the patient's eye. This is 6 months later, right? Of course, there has been refinement that has been done to this surgical method, but this was born out of this idea, was born out of a thought experiment that somebody that we had when we had our combined meeting with our collaborators. So from this part of it, this was 3 years, and there were some very good papers that came out of it. So this was a very highly productive and a very well advertised work. We got the funding from Wellcome Trust. We got, a lot of uh, what do you say, advertisements in papers and stuff like that. And the very fact that a new form of surgery, which was less costly, which was less cumbersome, and can be done by surgeons world over without the need for a manufacturing lab itself was a big deal. And the SLET was the biggest output from this entire project, right? So I learned personally to take a product from the lab to the clinic. So this was a huge learning experience for me. Most importantly, your mentors. Your mentors are very, very important. I had two wonderful mentors, Dr. Sangwan and Dr. Sheila McNeil, and they were, real gurus and teaching us the right way of doing what we have to do in the shortest period of time. And it does take a village to do good work very fast. So you can see all the faces that are on the right. These are all the people who worked on the project continuously for 3 years. We got about 12 papers out of it, and we did a lot of conference presentations, etc. An interested clinician has to be on your side, and that is the deal breaker. If you do not have an interested clinician on your side, your product will never see the light of day. And importantly, every idea is worth pondering. So if Dr. Sangwan had not slept on the idea of directly transplanting these onto the patient eye, SLET would not have been born at all. And right now, SLET is being used worldwide by so many clinicians, even from areas where they do not have access to clean room facilities. All right, so that brings me to the last phase where I became an independent researcher, and this happened in the year 2013 when I joined LV Prasad as a research scientist. So I decided by then that I wanted my lab to work on the corneal endothelium because I felt we did not know enough about these cells. So my lab right now works on all these aspects. So we work with the eye bank to improve preservation methods so that we can increase the lifespan of the donor tissues that are, donated. We also look at clinical translation. So we actually were able to generate clinical grade cells from donor tissues, which can go for transplantation. We also work on pathobiology. We want to understand some of the molecular mechanisms that are involved in disease conditions such as Fuchs dystrophy, PPCD, posterior polymorphous corneal disease, and then eye syndrome. So we are looking at the pathobiology, the mechanisms driving these disease conditions. We are also working on engineering the endothelium for transplantation. So we are using silk fibroin, silk from which is used for making our silk saris. So we are using the silk fibroin material for culturing these cells for transplantation. And we are also working on understanding very basic fundamental biology of the endothelial cells. We want to understand, say, for example, how do the cells sense and respond to pressure changes? When we talk about intraocular pressure, we only talk about the trabecular mesh cells. However, we also know that the endothelial cells are affected, not to the extent of dysfunction, but we know that their cell density reduces. So the cells are able to adapt until the time they die, but what makes them adapt? How are they able to sense this change, and what mechanisms drive that adaptation? So these are some aspects that my lab right now is working on. So um, these are my PhD students. One is working on what we call as trip ion channels, and the other who worked on the silk for Brien, she has now graduated and is in Chapin's Harvard doing her postdoctoral fellowship. So I will not bore you with the details of the science itself. So again, in this journey since 2013, now 12 years down the line, I have learned that research is a very, very slow process. So you have to keep yourself self motivated. And if you are going to do research in India, especially this kind of research, then motivation, keeping your motivation up and determination to see your project through is huge, because compared to the US or elsewhere, it takes a long time for you to get materials to do your stuff. Um, what I tell my students is whatever you write well and you contribute meaningfully to science. I don't believe in salami publications. What that means is you take one part of your work and you present it as a paper, and then you basically chop up your work into multiple slices so that your quantity increases, but the quality is not so good. So in our lab, it is they have a decent, comprehensive piece of work before they can publish, and we work with integrity and we try to work smartly, trying to keep our motivations up. So this is my lab. We get funding from the government for conducting our research. So in all of this, how has optometry helped me? You guys might be wondering, although I'm an optometrist by training, has it really played any role? It has, because usually when a basic scientist and a clinician talk to each other, a lot of it is lost in translation because they don't understand each other's perspective. So this is one area where I think I have been able to sort of talk to both ends. There are some questions that a clinician has asked, and we have addressed it in the laboratory. So now clinicians come to me and they say, okay, I saw this in a patient. And so what do you think is happening? And now I don't have to scratch my head and ask, what do you mean by this disease, right? So that basic training in the clinic has been a major advantage. It has also, the training in basic research has helped me address clinical questions in a rigorous manner and effectively bridge the gap between these two. So I would like to leave you guys with this quotes. And so I am open for questions. I believe we have another 10 minutes.
Thank you, ma'am, for the wonderful and inspiring presentation. It was so interesting to learn more things from the presentation and
Thank
from
you.
your talk.
If anybody has a question, you can.
Can I ask a question, Shamla?
Yes.
There is a question from Priyadashini.
Yeah.
Maybe you take that, and then I'll ask.
It's from Priyadashini. It was very surprising to know about your journey by integrating optometry competencies with biochemistry. I'm very curious to know about the challenges that you have faced and how did you overcome when you entered the basic science stream in research?
Like I said, it was not easy. There were many days when I asked myself why I am doing this to myself. However, persistence. I mean, I guess also probably because I like these challenges, and I like to solve puzzles. So to me, research is a puzzle. The questions that we ask are puzzles. How did I overcome? How did I sort of? So I guess the question is, how did I learn biochemistry or biotechnology skills, right? Like I said, you basically take every opportunity to do whatever technique that you want to do, because one is theoretical knowledge, the other is practical knowledge. And unless you do something, you will not know whether you're good at it or whether you got it right or what mistakes you're making. So I had to I had to Google search every single word. Like I said, I used to sit and read a paper 20 times till I understood what it meant, what it was conveying. And I used every opportunity available to do work for others. So that way my skills improved. So these were some of the ways, and I used to read a lot of the basic biochemistry books. And, I did attend a lot of basic coursework at Indiana University, which we were allowed to do. So I learned statistics, biochemistry, genetics. So I used to go and sit in on the 101 classes. And these days, I think you have a lot of YouTube universities. So these days, if I don't know something, I go to YouTube and I search. And I hope that answered your question previously. Okay.
Thank you. Thank you, Dr. Charanya. Am I audible and clear?
Yes, yes, Gopi.
Yeah, so thank you so much for accepting the invite, especially during the quick conversation. As I ended up in meeting you in New York, we just immediately accepted. And I'm curious to know, like, many of the basic science researchers end up in academic research like you, like maybe in as a postdoctoral researcher or in where they have more of clinical work as well. I want to know is the industrial scope if they land up in basic science research doing a PhD like you. Is there a scope in industry research, maybe in like any in contact lens industry or pharmaceutical? How is the intake? Because there will be a lot of competition because there'll be equal number of people who might have done basic science research and competing along with you. In that, since you are from optometry, does it reduce the chances, or how is the scope?
So you're asking whether it reduces my chances of getting into the industry or into academia?
Industry. Yeah,
Industry.
maybe you can address both of them. Yeah.
So I would say actually there are more people who have gone into the industry than the number of people who have stayed in academia, because the academic positions are far and few, to be very frank. So the competition for academic positions is more, I would say. That is not to say that industry positions are easily available. So in both, unless you are really setting yourself apart as something, you have a unique selling point, unique skill sets to add to that particular requirement, neither academia nor industry is going to take, right? So in terms of the overall scenario, I think in India, the academic scenario is a little better than the industry scenario, primarily because not a lot of research and development activities are taking place in India. That is primarily done elsewhere. And we are more of the clinical trial, centers and, those kinds of things. Having said that, you really do not require to be in a position for you to be doing research, right? I mean, in the sense you have your patient, you've seen this, you simply go back and, understand what you saw in that patient. That itself is research. And if you are seeing a bunch of cases, you can, sort of put together a case series. You can start your own research, which does not require too much of overheads as well. But I guess that's sort of an aside. But in terms of positions, I think right now you have equal competition in both. There is no easier, game out there. But what will set you apart is, acquiring skill sets, understanding what the requirement of the environment is and acquiring those skill sets. So, yeah,
Thank you, Dr. Charanya.
sure.
Thank you, Dr. Charanya, for the wonderful presentation and the talk, for mostly spending the valuable time with us today. So, before we wind up, I'll post today's quiz link in the chat box. To fill it to get the OCA certificate, which is signed by both Acchutha and OCA. To fill it, thank you, Dr. Charanya, and everyone for joining us today for the session.
Thank you. Thank you, Saranya. Thank you, Shamla. Thank you, Gopinath, for, some sort of inviting me and hoping that somebody, at least one person, is inspired to sort of, take a different route and try it out, whatever it is. All right. Thank you very much.
Thank you.